Hi everyone, I'm Yi Wang from National Tsinghua University. Today I'm going to share our work about a small molecular compound that can be a potential candidate to treating traumatic brain injury. First, I'll begin with a short introduction about traumatic brain injury, also known as TBI. WHO has estimated that around 69 million people suffer from TBI worldwide every year. The neuron death, BBB permeability, and inhibition of microenvironment makes it harder for patients to recover from TBI. Current treatment of TBI can be divided into acute phase and non-acute phase. In acute phase, surgeries may be performed to stop the bleeding or remove the blood clot, also with some anti-epileptic drugs to prevent possible epilepsy after TBI. In non-acute phase, rehab was used to support the recovery of patient. However, there is still no proactive therapy that can promote neural regrowth and functional recovery in clinical. Therefore, my work aims to find out a small compound as a potential candidate to be applied in clinical. After a series of screening, isoroifolin was identified and the efficacy was examined. As a result, isoroifolin demonstrated a high efficacy and wide-range treatment window on promoting neural regrowth of injured neurons. In addition, the toxicity assay also indicates a good safety profile. Furthermore, the PK analysis also confirmed the penetration of isoroifolin to brain. The underlying mechanism of isoroifolin was also being addressed. We found that isoroifolin binds to S1PR3 and inhibits the phosphorylation of CK2 and GSK3 beta, leads to increased expression of regeneration-associated genes. Also, the inhibition of S1PR3 through non-ligands and isoroifolin were shown to increase neural regrowth. Furthermore, in proteomic-wide screening of the target protein, we found that in glial cells, isoroifolin also binds with NQO2 and decreases the cytosolic and mitochondrial ROS after injury. In conclusions, we found isoroifolin targets S1PR3 to promote neural regrowth in neurons. In addition, it also binds with NQO2 and decreases inflammatory response in glial cells. For histological validation, isoroifolin successfully increased the neural regrowth in 3D brain slice model. In cultures in mouse TBI model, isoroifolin also enhanced tissue repair more than 50%. For in vivo experiment in horizontal bar test before injury, the mouse in different groups showed a similar motor ability. After TBI, compared to Shen group, the DDH12 group showed a significant motor deficit until 6 dpi. With isoroifolin treatment, the TBI mice showed a rapid function recovery on 1 dpi. A similar result was shown in the Rodarot test. The DDH2 group experienced motor deficit after TBI, and isoroifolin enhanced motor function recovery up to 35% rapidly. That concludes my presentation. With further research, this study may offer new hope for recovery and improve outcomes for TBI patients in clinical. Thanks for your attention.